So we are going to wrap up chapter 2 here. What I want to do is go back and recap the mitochondrial structure before we move on to the other non-membranous structures of a cell. So let's go back. What is what I can refer to today without giving the name away, this first layer of the mitochondria? What do we call it?
Outer membrane.
The outer membrane. And the second membrane is referred to as the
Inner membrane.
Inner, very good. What about the space between the two membranes? What do we refer to that as?
The intermembrane space.
Intermembrane space or fluid. Intermembrane space, I think, is more common, but there is fluid in there. But the key thing is the inter, I-N-T-E-R, meaning what? Between. Okay, so it's the space or the fluid between the two membranes. And the innermost fluid-filled area is referred as the what?
Mitochondrial matrix.
The mitochondrial matrix. Very good. The innermost fluid is referred to as the mitochondrial matrix. I try to give you guys a little bit of heads up. Last week, that matrix is a generic word, and so I'd like to go ahead and precede that with mitochondrial matrix. That gives you some more specific location.
So the folds are what you would call the cristae, right?
Correct. The inner membrane folds into these things called cristae or cristae.
Okay.
And so why do we want these mitochondrial inner membranes to fold into cristae? To increase surface area. We can get more operation in a smaller space. So we're just increasing the surface area. Where we're transitioning to this week in chapter 3 is we're going to see physically why we need that increased surface area. There's going to be a lot of movement across the inner membrane between the matrix and the intermembrane space, back and forth. We're going to be moving some protons and electrons back and forth. Did we draw anything on our membranes last week?
Proteins.
We drew some transport proteins where?
All over there. On the folds.
So on the inner membrane. Very good. So let's go ahead and draw some, if you haven't done so on your mitochondria, draw some transport proteins on the cristae of your mitochondria, of the inner membrane. And these are going to be various types of transporters. We're just introducing them now. We'll look at them in a little better detail in chapter 3. But we're trying to acknowledge that there are transport proteins that physically carry protons and electrons between those two areas. Did we acknowledge any protein in the outer membrane? No. There are proteins in the outer membrane. So why don't you draw some pores? Does that word make some sense? A pore would be type of what? Type of what might a pore? How can we do a channel? Okay, so draw some channels. You don't have to put a bunch, but maybe a few pores. P-O-R-E, pores in your outer mitochondrial membrane. I'm going to put a little protein embedded in this outer membrane. So it tells us the outer membrane's porous, means particles can get in and out of mitochondria, which is very useful. And then we'd find many more transporters in the inner membrane when compared to the outer membrane. All right, so the outer membrane is referred to as being porous. Inner membrane has transport proteins. This image might help you a little bit complete your drawing which you were looking at. We can see the cristae folds. The purple things are representing the transporter proteins in the inner membrane, and you contrast that structure to our pores. They're called corons. The name's not too tricky there. Okay, they're porous proteins. So let specific particles from the cytoplasm to the what? From the cytoplasm to the intermembrane space. There you go. But if something needs to be transported from the intermembrane space to the matrix, we probably need a transporter. Good. Additional or any questions at this point about the basics of the mitochondrial structure? We will put this structure to use coming up, chapter 3. Okay? But that, discussing the mitochondria concludes our discussion of membrane-bound organelles. However, when we talk about what is inside of a cell, we can't omit non-membranous structures. I just kind of like the membrane limitation for an organelle. But there are other non-membranous structures in a cell that are very critical for this cell, a cell, to do its work. And the first most notable non-membranous structure in a cell would be a ribosome. What is a ribosome important for? You can use your handout that just came back around. What do ribosomes do? What's their job? Protein synthesis. They're there for translation. Our ribosomes carry out translation. In our narrow definition, we wouldn't include a ribosome because it doesn't have a what? A membrane. But please don't misconstrue that this thing isn't important or found in cells. It's critical for a cell to survive. It just doesn't have a membrane around it. So you might know what a ribosome is. We know what it does. What is it? It's what? Let's be a bit more specific. There is some ribosomal RNA in there, so part of it's nucleic acid. What's the other bulk of it? The what? The RNA acts as enzyme. Okay, so keep going. It's protein. Okay, so the ribosomes are kind of unique in that the enzymatic portion of the ribosome is actually the nucleic acid, the ribosomal RNA. But like other enzymes, the bulk of it is a protein. Okay, so your ribosomes are a complex of two proteins with some ribosomal RNA in there. This is a conglomerate, if you will. It's a protein with some RNA in it. And as noted, our ribosomes are analogous to enzymes because they do carry forward translation, right? They catalyze, right, that protein synthesis. So they're very much like an enzyme. And technically they are. Where do we find ribosomes? You may find them in the cytoplasm or on the membrane of which organelle? The ruffyar, and there's no synthesis going on with the Golgi, so you wouldn't find them in the Golgi. You would find them on the ruffyar. Do what? Is there ribosomes in the mitochondria? They're not on the mitochondrial membrane. I'm having a complete purge, but I believe they are in the matrix as well of a mitochondria. But you would find it on the membrane of the mitochondria. What other membranous organelle will we find ribosomes? The what? The nucleus. Do y'all remember seeing that when you study the nuclear structure? You'll find ribosomes at the nuclear envelope. You'll find them most notably at the ruffyar. They're also free in the cytoplasm as well. Okay, so there's some different locations where we can make proteins. At the very end of this guided note discussion, we're going to differentiate protein fate, whether the ribosome is associated with the ruffyar or in the cytosol. Okay, so we'll come back and differentiate those. Before doing so, we need to talk about some other structures inside of a cell. Collectively, there's a set of proteins which make up what's referred to as the cytoskeleton. The term cyto refers to what? Cell, the skeleton, the scaffolding, or the structure. So we have a protein structure inside of our cells. The diversity of the cytoskeleton is dependent upon the type of cell. So not all cells will have the exact same cytoskeleton mixture. The cytoskeleton of a nucleus, of a nerve cell, would be different than the cytoskeleton of a muscle cell, which would be different than the cytoskeleton of an epithelial cell, for example. The cytoskeleton is just a mixture of different types or families of proteins. So these are cytosolic proteins. These are proteins inside of the cell that have a specific shape or a specific what? Function. Very good. The three categories of our cytoskeletal proteins include microtubules, microfilaments, and intermediate filaments. As you notice, maybe through this class so far, that you can't just kind of take the prefix of something and run with it. You need to evaluate the entire term so you don't get something confused. A microtubule is very different than a microfilament. The tubule forms what? Microtubule looks like a tube. A filament looks like a line of fiber of filament, right? And an intermediate filament also looks like a filament, a strand. Microtubules, anatomically, in comparison to the other three families of cytoskeletal proteins, are the largest in diameter, and so they are physically the largest. And this is backed up with the fact that they actually make a tube. And so they're big enough to make a big, large structure, and it looks like a tube. I have a note here that the cytoskeletal, the microtubules change often. What I mean is the microtubules can grow, using the term loosely, they can extend and shorten very quickly. They can assemble and disassemble, or the proper term would be polymerize, depolymerize, very quickly. They can be built up and broken down really quickly. You can probably appreciate this when you study your Gen Bio class and you talked about cell division and how this the mitotic spindle formed and pushed the chromosomes to the middle of the cell and pulled them. Okay, so we can build and break it down fairly quickly. So the microtubules, they do form tubes that can build them quickly or break them down quickly. Where we're going to see them is serving as a network for transport within the cell. A network for, let's be more specific, vesicle transport. Our microtubules are useful for transporting vesicles, for example, from where to where? Where do we see vesicles transported? From which organelle? From, back it up a little bit. Say from the Ruffyard to the Golgi, or the Golgi to a certain part of the cell membrane. That's regulated. We had a discussion about this. That's regulated, and what's used are microtubules. The microtubules are oftentimes described as being like a track, like a railroad track would be served for a pathway for a train, right? The microtubules would serve for a pathway for our vesicles. And that's where we're going to see these microtubules. Most notably, when we study the nervous system coming up in Unit 2, we're going to talk about the production of neurotransmitter. It's going to be wrapped in vesicles and transported from the Golgi all the way down the axon to the terminal. And if you think about some of your nerve cells, how long they may be, you heard of your sciatic nerve, right?
Oh, yeah.
Runs. So it's like personal experience about that. So it's a long nerve. And if you're tall, right, it's very long. If you're not very tall, it's not very long, but it's longer than a few centimeters. And so vesicles have to be shipped or trafficked along microtubules down that whole however long your nerve is. And so we use the microtubules for that transport. What's cool about the microtubules is that they're shipping and receiving kind of all the time. So it's called trafficking. Just like your cars on the highway, vesicles move back and forth along the microtubule network. Let's add one more note about the microtubules And that in order to do this trafficking, they use motor proteins, M-O-T-O-R, motor proteins. And I'll have a visual for you in just a moment about a motor protein. So that's an introduction to our microtubules, the largest in diameter. They can build and disassemble pretty quickly. On the opposite end of the size spectra are the microfilaments. These are actually the smallest in diameter, and they are more stable than the microtubules. What that means is once they're built, they're probably going to stay built for a period of time. They're not going to just, I mean, they can assemble and disassemble, but not at the rate like you would find a microtubule building and breaking down. So your cell can make a microfilament bigger or shorter or whatever, but just probably not as fast as you would see a microtubule building. These also associate with motor proteins. So they are used for some type of what? Transportation, movement. We're going to see these most specifically in our muscles. So how your skeletal muscles, muscle contraction of all of your muscles, your skeletal muscle, your heart muscle, and your smooth muscle needs these microfilaments associated with a motor protein to get some overall movement. And in this case, we're looking at the cell. That's one example of what these do. A third type of cytoskeletal protein family are classified as intermediate filaments. And these kind of just got lumped in the middle. We have the biggest, the smallest, and now the ones that are size-wise, though, intermediate is in the middle, right? So they're not the biggest, they're not the smallest. Diameter-wise, they're intermediate between the microtubules and the microfilaments. But as noted, they are the most stable, meaning that once they're formed, they're going to be the least likely to break down, destabilize. They may, but they're going to be less likely. I mean, they may persist for weeks or something like that, or longer. The intermediate filaments give physical structure to us, to wherever it is. And so you'll find these in great concentration if you think about parts of your body that undergo physical, mechanical stress, like your skin, for example. That's some intermediate filaments in those epithelial cells to give them some physical structural support. And we wouldn't want that support to just dissipate randomly, right? And so these things are just stable, they'll last a while. You're probably most familiar with one family of common example of our intermediate filaments is called keratin. You've heard of that one? That's an example of an intermediate filament. We hear about it in our beauty products, right? The hair and the skin and etc. Right? So it is a stable protein. So those are primary differences between the cytoskeleton. A big thing to acknowledge is that these are intracellular proteins. And their diversity depends on the cell type. If we look at an image to help represent that transport, what type of filament is this? Microtubule. I can see the tubule. If you look at it, it's got a hollow inside. And the motor protein would be these purple things. And the vesicle is also colored purple in this image with a little blob. But the way the vesicle gets transported is using these little foot or motor proteins. So cell biology, we talked about, here in cell biology, we talked about quaternary level. The big tube would be the quaternary. Each little tiny protein, the red or the yellow or whatever, would be a tertiary level protein. So that means we're just adding little full proteins to the end or taking them off, right? So the ends can grow and break down by adding more or less proteins to it. What is the yellow? What type of? A microfilament. Very good. You can also see some transport, but we'd also see it more with, we're gonna see it with contraction. Part of. A microfilament also represented here with the yellow. It's just kind of turned on its end. So the microfilament would be here, the motor protein also represented in the purple and the red. That physically just interacts. The mechanics, we will review this when we look at muscle physiology in Unit 3 this semester. So in a couple of months, we'll look at physically how that happens. Right now, we're just introducing these proteins and what they're generally used for. And if you recall back to the beginning, okay, a couple of weeks ago, we talked about cells joined with like cells can form a what? A tissue. To join them, we have to, some tissues we have to physically affix together using specialized proteins. And that joining of the tissues together, or cells together, excuse me, are called intercellular membrane junctions. So let's work through this backwards. This is a, how else can we describe that? It's a junction, joining, right, connection of the membrane between cells. So we're just joining the membrane of different cells using very specialized membrane proteins. A week ago, we would have just said integral membrane proteins. Today, we're looking at them complex in these big structures, and we give them names and major functions. We're looking at some of these integral membrane proteins specialized to form these different junctions. There are three types of junctions which we will study. This is certainly not an all-inclusive list, but these are ones that we're going to see this semester. But if you look at this, we have three entire cells represented in the portion of a cell. And if you look at their membranes, we can see some little protein networks. So we're going to zoom in and look at those junctions. The first type of junction is referred to as a tight junction. And again, the nomenclature is pretty descriptive. What do you think is happening at a tight junction? They're brought in closely, right? So the protein, the membranes are joined together, closely associated. So the proteins that join their membranes form a tight junction, or basically a barrier. We haven't talked about transport mechanisms yet, but we will in chapter 4. So a tight junction, let's think about it. What's the probability of a molecule, say like sodium ion or water, spontaneously just diffusing from the cytoplasm of one cell to another where there is a tight junction, where we have not one but two layers of phospholipid bilayers sandwiched? Is it high or low? Very low. Never say never or always, but the likely is extremely unlikely. The likelihood is not there. And so where there's a tight junction, what are we doing? Closing off that section. For what reason? Why might we just close off a section somewhere? These don't give quite, I mean, they will give some strength, but not a lot. And so more protection to prevent what? Something from moving, right? So it's like a physical barrier to prevent any molecules from diffusing from one cell cytoplasm to another at that location. And we're looking at specifically where that is. The probability of something simply diffusing is minimal. And therefore, you would find tight junctions in tissues where you wouldn't want a lot of cell-to-cell diffusion, like in your epithelial tissue. The lining tissues, or those barriers that we have. Another type of membrane junction that we'll see this semester are referred to as desmosomes. Desmosomes structurally I don't know if this is a proper term or not, but I'm gonna say are more diverse. We're contrasted to a tight junction. And what I mean is the membrane proteins of one cell lace out and interact, intertwine, bind with like proteins of the adjacent membrane of another cell. Visually, kind of analogous like we did when we stuck our hands out, right, last week. We did our little protein receptors, like that, but the interaction is permanent. Kind of like Velcro. What might Velcro do between two cells? Physically bind and do what? Stick, right? So, at a desmosome, we're binding to membranes, we're adhering them together, and oftentimes you'll find them on the intracellular side attached to a cytoskeleton. And so it's almost like a cable network that it attaches to on the inside. So what it does is physically distribute tension. The desmosomes distribute physical tension. So it's not at one localized spot. I just spread it out. Why might we want to disperse physical tension? In what fashion?
Equilibrium, and you kind of like you feel inertia, you push against the seat, you feel that strength being used.
Okay,
And
so,
that was something similar in the cellular level.
okay. That's good. I've never thought of it in using that term balance, so that's why I'm just kind of thinking. So very good. We could use that word.
There's control, maybe.
Doesn't really control per se. I want to be careful using that. But think about tissues and think about your heart. This is where we're going to see these next. Your heart. What is your heart always doing, hopefully? Pumping. Squeezing and stretching. Contracting, right? Force, stretch, force, stretch, force, stretch. Without desmosomes, what might your cells do? Force, stretch. Even before then. Because they're going to tear apart, right? If you're just continually wrenching on something and stretching it, and you have these tiny little cells, and we didn't have them interlaced, they're probably going to rip apart. And a ripped apart heart, is that good?
No.
No. So let's prevent that. We want the heart to generate the tension, right? But we don't want that tension to be localized in a single spot where we can pop the multiple cells apart, cause a heart attack. That's not good. So we find desmosomes between those contracting cells in the heart to distribute the tension. So it's not at one spot, but it's spread throughout. And so throughout, it's kind of like making the force load light and distributing it among many spots instead of just one big location. Does that make a little bit of sense? Tension distribution there with the desmosomes. There are variants of the desmosomes, adherents, and etc. But for right now, we're just going to categorize those. A third type of junction are referred to as gap junctions. These two are also very descript. These form a what? Between the two cells? A gap. They basically look like what? They look like channels, but it's not through one membrane, it's actually through two, right? So the like protein is expressed on the neighboring cell, and they're aligned to one another to make an entire channel. Why might we need a channel?
Let stuff cross.
To let stuff cross from the cytoplasm of one cell into the cytoplasm of another. Feeding off of that, if I need a gap, basically, what type of particles might need a channel if we want them to move? We want them to move from the cytoplasm of one cell to another. Proteins are going to be a bit too big for a channel. Because if we let a big protein in, probably everything could go through. So let's back it up size-wise. Ions like sodium or sugar is also going to be too big. So, I mean, That's good. I'm not discounting you, I'm just gonna clarify. Do I have it? Potassium, water, so smaller like ionic things, so tiny.
Is it always ions?
As far as I know, yes, but, you know, I don't know everything, so. I don't think there are, I really don't think there's aquaporins, but technically, they're moving solute through, you can probably, a water could probably go through. But I don't think there's a gap water junction. They're usually specific to ions, sodium ion, potassium ion, calcium ion. Where we are going to find these are in cells that electrically need to connect. We need to electrically connect some cells, throw in a gap junction, zip some ions through there.
So it would just be like a series of channels between two cells.
Very good. You said a series of channels between two cells, but they're aligned.
Yeah.
You're going to find this in your heart, so the entire organ can work together. You're going to find this in some smooth muscles, where they all need to work together, like in your digestive system. So it can conduct electrical impulses through really quickly.
Where did you say it located?
We're going to look at them in the heart and some smooth muscle. So those are our membrane junctions. Go ahead and go back and review the specificities, and if you have questions, right, bring those to class next time. That actually concludes what we're going to talk about as far as the internal structures of a cell. But as I mentioned, I want to go back and differentiate proteins, whether they're manufactured at the rough ER or in the cytosol. So let's review that. I have a similar image in your guided notes if you're using it, but it's not exactly the same one. So I'm going to add some detail to your guided notes image. If you recall, hopefully you recall from our discussion last week, we learned that the rough ER manufactures proteins. We also clarified that the proteins when manufactured actually get dumped, if you will, in the lumen of the rough ER. At first it maybe sounded a bit confusing until we added that the rough ER actually buds off little things. What do we call those? Vesicles. Those vesicles we now know can be transported using microtubules to the Golgi, where that protein can be modified, its carbohydrate groups change or whatever, and then the Golgi can then bud off its own vesicles with proteins. Here's where we clarify though. If a protein is originally manufactured at the rough ER, its protein is going to have one of two fates. What were the two fates of proteins manufactured originally at the rough ER? It's going to be what? Secreted as like a messenger, like a hormone or neurotransmitter. So we said the two fates is one, it may be just physically secreted if the protein is what? Solubility-wise? 50-50 not it, so hydrophilic. If the protein is hydrophilic on the outside of its water soluble, this will come into chapter 4. If we exocytose it out into the environment, if it's hydrophilic, it's not in that vesicle membrane, so it just goes out into the extracellular. The terminal diffuses out in the cytoplasm. So, if a protein manufactured at the rough ER, modified at the Golgi, if it is completely right at the surface hydrophilic, that thing will be secreted. That protein. It's going to be a messenger like, say, insulin, right? You've heard of that. We've been using that as an example in the last couple of weeks. It's a hydrophilic protein, so it gets released out into the extracellular environment. Some of our neurotransmitters from our nerves, they get They're hydrophilic and they get released this way. However, we learned that not all proteins are strictly hydrophilic. Some are amphipathic. Those proteins that are amphipathic, even though they're going to the membrane, they aren't free. And so what happens to those? They get incorporated into that membrane to act as and fill in the blank any membrane protein we've studied. Form a gap junction, form a receptor, form a channel, form a pore, form a whatever, fill in the blank, all the things. And that's where our membrane proteins come from. They originate where? At the rough ER. So the rough ER manufactures proteins that will go where? Either secreted out into the, from the cytoplasm to the extracellular. So our proteins in the rough ER may be secreted extracellularly. Or they may be, if they're amphipathic, they will be embedded or incorporated or integrated into the membrane. Serve as an integral membrane protein. Okay, so far? So we have an idea how proteins can get outside of the cell, or proteins can get in the cell membrane, but we haven't acknowledged about proteins that would work inside of the cell, like to form a microtubule, or an actin filament, or an enzyme, or pick your favorite intracellular protein, right? We haven't acknowledged how those are manufactured. Those are manufactured not at the rough ER, but where? In the cytoplasm. And this is what you'll want to add to your image. Okay, so just draw a ribosome somewhere in the cytoplasm of your cell. You can have it manufacturing a protein. You can make a protein just a line. In this image, the ribosomes are the purple things, if you can see them. And then the protein that's being manufactured is the little blue things. And so when the ribosomes produce proteins in the cytoplasm, those proteins are going to stay in that cell. Would they have a membrane around them? Would cytosolic made proteins have a membrane around them?
No.
No, because they aren't made in a membrane containing something. There's no source of membrane. And so they would be what you would refer to as dissolved in the cytoplasm. So reminding you as well, some examples of cytosolic proteins would be your cytoskeleton. Each of those, the tubulin, the actin, whatever. If they're going to work inside of the cell, they've got to be made inside of the cytoplasm. Enzymes, etc. I see some scowls. So what questions do you have about the difference between proteins being manufactured in the cytosol versus the rough ER?
What was that term you used for the ones that are in the cytosol?
Dissolved.
Oh, just dissolved.
Dissolved is the term I use. You could say dissolved, you could say free, but they are not membrane bound. This looks like this concludes our Chapter 2 discussion. Are there any questions at this point before I move on to Chapter 3? All right, we're going to go ahead and transition now to Chapter 3, which is dealing with metabolism. I've actually kind of stripped it down to the nitty-gritty. Metabolism is something that you're going to see probably in, if not all, but most of your biology classes. You'll introduce it in gym bio. You'll talk about it here. You're going to talk about it in micro. You're going to talk about it in cell, botany, right? Pick all your favorite classes. You're going to talk about metabolism because metabolism is critical to cell survival and to this energy transformation. If we review the objectives, which we're going to cover for Chapter 3, we're going to just give a good definition to metabolism, and we're going to differentiate a catabolic and an anabolic reaction. And then we're going to differentiate a hydrolytic or hydrolysis, condensation, phosphorylation, dephosphorylation, oxidation, reduction reaction. Some of those may be familiar, but we're going to review some of them to make sure we're okay with actually using the terminology. Then we're going to go on and add the structure of ATP. And yesterday I added, so in your if you've got your guided notes objectives, in addition to the structure of ATP, we're going to talk a little bit about NAD and FAD. So in objective 3, add also NAD and FAD. We're going to talk about what they have to do with metabolism. And then not today, but hopefully on Thursday, we're going to summarize some major metabolic pathways, including glycolysis, this thing called the intermediate or the linking step, and then the Krebs cycle and the electron transport chain. So last week I gave a plea to you to go ahead and read chapter 3 in advance. If you've not done so, it's going to be critical that you do so before Thursday. And there's some videos, supplemental videos that will maybe a more passive way to get into the material first. So watch those. If it helps you watch those maybe before you read the textbook and get into the nitty-gritty, I would do that. And then at the very end of chapter 3, we're going to acknowledge that not only carbohydrates are used for ATP synthesis, but also fats and proteins are major dietary sources of ATP. And then we're going to go through and talk about differentiating a lytic versus a genesis type of reaction. So if you look at some of these terms, they look confusing. If it goes, we'll get familiar with the terminology so we don't get them confused. We're going to compare glycolysis versus glycogenolysis versus gluconeogenesis, glycogenesis, lipolysis, lipogenesis. Hopefully you can kind of see the oppositeness between those, and proteolysis as well. Again, we probably won't get to items, we will not get to items 5 or 6 today, but hopefully on Thursday. On your own, if necessary, okay, so I won't hold you directly accountable for the different types of metabolic pathways, section 3.1. Metabolic reaction and delta G and all those things, 3.2, or reaction rates, 3.3. That's background information. I'm going to carry on from there. And so if you need to do that background work, do so. I do want to go ahead and make sure we are okay with some basic terminology with metabolism, which we'll get to in just a moment. But before we get to the specifics, I would like to just kind of acknowledge what a cell might need actually energy for. And we can super simplify our list of what cells might need energy for into a list of 3. Again, not all inclusive, just kind of giving you an idea. Why might a given cell need energy? To synthesize molecules. I'll call that chemical work. So cells need energy to carry out chemical reactions. So some chemical reactions require an input of energy, like synthesizing proteins, like synthesizing ATP, like synthesizing a lipid, right? So on and so forth. Okay, so some chemical reactions require an input of energy. What else might a given cell need energy for besides chemical synthesis type work?
Mechanical.
Mechanical work, very good. It's like y'all knew what I was going to say. We can also carry out mechanical work. What would be an example of mechanical work? Cell division takes energy. What else? Transportation. Did you say replication? Of the cell, that would be cell division, replication of the genome. It also goes into, it does take energy. What else is mechanical? So some organelles may need energy to carry out their, so think mechanical. Do what? Heart to do what? To pump, right? So a contraction like your heart muscle, contraction like your skeletal muscle, contraction like your cell muscle, smooth muscle cell, excuse me. Mechanically, using those motor proteins, for example. Those motor proteins are going to take energy. Good. Adding? Very good. So cells need energy to do chemical work, they need energy to do mechanical work, and they also need energy to sometimes transport molecules across a cell membrane. So different types of cell transport. We'll identify in chapter 4 as active membrane transport is going to utilize energy as well. Again, this is a really condensed, let's just kind of give an idea. Most things that a cell is going to do is going to require an input of energy. Not everything, but quite a bit of any cell action is going to need energy. So if a cell wants to live, it's going to need energy to do its various types of work. In order to understand how this happens, we need to look at some basic definitions. Okay? The first thing is, let's just define metabolism. How can we define, let's define metabolism. What is metabolism? Okay, that's half of it. You said the breakdown, but it's also the
Total of all chemical reactions that occur in your cells.
okay, so very good, the total. Not only breaking down, but also
Building up.
building up. And so an easy way to say that is the total or the sum of all biochemical reactions in a cell. So it's inclusive. So metabolism is just all the biochemical reactions occurring in a cell, or you could look at an entire organism, person. The more famous metabolic reaction is identified is those that are called catabolic reactions. A catabolic reaction, what happens?
Breaks down.
We can use the word breaks down, right? It takes a larger substance, breaks bonds, and the resulting substance is smaller. Fat catabolic. Right? A fat catabolic reaction does what? Breaks down fat to release what?
Energy.
Energy. So your catabolic reactions, if you will, break down, but they do release, my abbreviation here is E for energy. And thermally speaking, they're referred to as exergonic, meaning the heat is going which way? Out. So these release energy. Catabolic reactions take larger substances, make smaller substances, and in doing so, energy is released out. You will generally see a catabolic reaction with the suffix lysis. So hydrolysis, lysing, right? We're splitting. So if you see the suffix, that molecule, if you see the suffix lysis, the molecule is being what? Catabolized, broken down to release its energy, any stored potential energy in it at that particular bond. Conversely, some reactions are anabolic. What happens in an anabolic reaction? You form bonds. So, what does that mean? Yes?
Store energy.
You create a larger molecule, and in those bonds we are able to store energy. So we're able to store energy. So oftentimes it's to make the bond we require some input of energy. So these actually require an input of energy. And therefore we need to put heat in or energy in. So you would refer to as endergonic. But the key thing about an anabolic reaction, even though it does require energy, it does then give the ability to store potential energy. So if I build a molecule, yes, I'm putting energy into it, but then I'm storing it for later use. So I'd refer to those as anabolic. You're probably more familiar with anabolic if you're exercising and working out, muscle building, right, shows, those kinds of things. You may take a protein anabolic supplement. That supplement will help you do what? Build specifically though what? Protein anabolic helps your cells build proteins. Okay, it's like protein anabolics reaction builds proteins. Okay, so it's like double saying it there. Hopefully the proteins in your muscle cells, okay, your motor proteins inside of your muscle cells. Bigger, bulkier, stronger. What about fat anabolic? What are you doing there?
Nothing.
Fat anabolic would be storing fat,
fat,
okay, in your fat cells.
What we don't want.
But some of us are really good at it. So, yeah, I got that anabolic reaction down, taking in the Twinkies. Okay. So anabolic reactions build molecules. So for example, we're going to look at ATP. If I want to build ATP, I need to carry out an anabolic reaction. I need to take ADP and add to it a phosphate to make ATP. But to make it, I need energy. But once I make it, I can store it and then catabolize it to release. And then once I've released energy, I can then hopefully
Do the process all over again.
do again, which we're going to do the next 2 days. So I'm excited about it. Yay, glycolysis, right? So we're going to go through some reactions. We're going to catabolize a molecule called glucose so that we can anabolize a molecule called ATP. Right? We're just transferring energy. And that's basic. Go back to your Gen Bio, right? Energy is always just transformed. And so we're just kind of taking it from one thing and giving it to another so we can carry out specific reactions. All right. So if we go through and look at these metabolic pathways, we've got to talk about enzymes, which we've used the word a few times this semester. What are enzymes?
Catalysts that speed up reactions.
Catalysts that speed up reactions. They're very specific. Most are protein in structure, with the exception that we talked about, the ribosomes, right? Most are just strictly just a protein structure. They're there to help a particular reaction carry forward, whether that reaction be catabolic or anabolic. They're going to have a specific enzyme. A lot of our enzymes may have also a coenzyme, different molecule, not protein, that is there to help a particular enzyme carry forward a certain reaction. And we're going to identify some important coenzymes. We're going to look at the structure of 2 of them, but we're going to talk about NAD, we're going to talk about FAD, and then on Thursday we'll talk about acetyl-CoA, another coenzyme. We're also going to talk about various oxidation and reduction reactions, so let's review that. Oxidation and reduction reactions have to do with what? Electrons. Just taking them and giving them, taking and giving. If a molecule has been oxidized, it has done what?
Remove.
Lost, removed, released. It's electrons. The electrons, if they are given up or oxidized from a molecule, another molecule has then just become reduced. So reduction is the gain of electrons. You may remember the old acronym, oil rig. Oxidation is lost, reduction is gain of electrons. And just quick reminder that when you first see that sounds confusing, how's the reduction a gain? We're reducing the charge because our electrons have what charge? Negative. So it's reducing the electrical charge of whatever's getting it, so you're reducing it. That's what we're going to do. Sounds terribly exciting. We're moving electrons. But if we keep it in a simplistic fashion, which I like to do so we can kind of get it, if I'm moving electrons, really what am I moving? To keep it simple. Energy. I'm taking energy from something and giving it to something else. So that something else can do what?
Use it.
To do work, right? To use it, do something. That's what we're going to do. Move electrons so we can do work. What we'll learn is that we're going to do it very I'm going to use the word loosely, slowly. All these reactions are fast. But we're going to do it procedurally so it's efficient. So when we're in the middle of the reactions, don't let yourself get bogged down. Think about, this step is here because it's efficient. As efficient as our body can be. Okay? So that we don't waste any potential energy. Okay? So a lot of molecules that we take in to consume, they have so much potential energy that if we don't extract the potential energy efficiently, that we might waste the potential energy in the environment as heat, which we don't want to excessively do. If you release too much heat, what's going to happen? What would happen?
Functions will slow down.
Or even just stop and die, right? So we don't want to die just because we ate a Twinkie bar, right? There's so much potential energy in there. But we want to be able to extract that energy more efficiently, release those electrons in a procedural way so that we can transfer it out efficiently. So the first structure, a coenzyme I want to look at is called NAD. I have a couple of, just found these images out there in the web. Looking at NAD, and label this first side the oxidized state. When you see NAD plus, it is oxidized, so it's given up its what? Electrons. If it's oxidized, it could probably also be reduced. When it's reduced, it interacts with hydrogen atoms. It can actually pick up kind of 1.5 hydrogen atoms. It can pick up a full hydrogen atom and the electron of another. And so in doing so, you can see the hydrogen atom, the extra one here. So NAD is reduced. When NAD has picked up hydrogen and a couple of electrons, you would refer to it as NADH. It is reduced. It has a little more what?
Energy.
Electron and energy, right? You're correct. Right. Energy. If it has energy, it could do what with it? Or, and it doesn't have to go back to the NAD. It could go to something else. It could transfer the electrons and the proton out to give something else the potential energy. So it can reduce and then oxidize, and then reduce and then oxidize, and then reduce and then oxidize and then reduce. That's what it does. It's referred to as a proton carrier. That's what it does. It picks up hydrogen ions and electrons and then drops them off. Picks up a little energy, drops it off. Picks up a little energy, and drops it off. Okay? So NADH is a coenzyme who can alternatively be oxidized and reduced, which is important for us coming up on Thursday. Very similarly, is a molecule called FAD. This one, when exposed to two hydrogen atoms, can actually pick both of them up. And so in doing so, it's going to be balanced, electrically balanced. So FAD, in this first box, we are going to see it. FAD is the, which form? Oxidized form. When exposed to two hydrogen atoms, we could then reduce it to form, we call it FADH2. And then it could be oxidized to release energy, and specifically we'll look at hydrogen ions and electrons. Very good. And then after it's oxidized, it could be pick up some more and be reduced, drop them off and become oxidized, pick up some more, reduce. Right? That's what these two things. They're there to carry electrons and hydrogen ions from one spot to another by just alternatively oxidizing and reducing. Does that concept make a little bit of sense without going to the detail of the structure? And another important structure that we're going to talk about is called ATP. ATP is a nucleotide. You would have covered that also in your Gym Bio class. And just a recap for you, a nucleotide is comprised of a base containing a nitrogen, a pentose sugar, and then also, in this case, we're looking at 3 phosphates. You would call this a nucleotide triphosphate. One, 2, 3. You can see the 3 little phosphate groups. It's referred to as a nucleotide triphosphate. It, anybody know the sugar in it? No pressure, Holly? Glucose? It's actually a pentose. Glucose is a, the option would be ribose or deoxyribose. 50-50 not